From Microbe TV, this is TWiEVO, This Week in Evolution, Episode 9, recorded on June 7th, 2016. This episode is brought to you by CuriosityStream, a subscription streaming service that offers over 1,400 documentaries and nonfiction series from the world's best filmmakers. Get unlimited access starting at just $2.99 a month, and for our audience, the first 2 months are completely free if you sign up at curiositystream.com/microbe and use the promo code microbe. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Utah, Nels Elde.
Hey, Vincent. Great to be here. I'm happy to report that summer has arrived in Utah. It's warm here.
Yeah, here too.
35°C for those of you keeping score at home.
That's a lot. We have 28°C and clear skies. It's supposed to rain, though, later today. So you're back in Utah, and last time I saw you, you were right here across the way on my desk.
Yeah, that was really fun. So I was out there, what was that, 3 weeks ago, something in that neighborhood now, for the Biology of Genomes meeting at Cold Spring Harbor, and had the really fun opportunity to sit down in vivo with you at TWiEVO World Headquarters. That was great.
It was a good meeting?
It was. It was really a fun meeting. Great energy there. And, I think genomics or kind of genome biology has sort of reached this point right now where a lot of the gene editing technology, so Crispr and related stuff, which we'll be talking about with our guest in today's episode, there are all these really cool sort of experimental torpedoes in the water as people are thinking about genomics and how this all functionally comes together. And so it just feels like we're at the beginning of a really fun time where these fields are sort of colliding, experimental biology and genome science in really cool ways.
Yep. Yeah. So we do have a guest. He is from the University of Texas at Austin. Jim Bull, welcome to TWiEVO.
Thank you. I look forward to this.
I met you a couple of years ago. I don't know if you remember or not.
I remember very well.
Okay. And
you told me about polio being loose in the sewers in Israel and a number of other things.
That's right. That's right. Well, we're almost eradicated in terms of polio. A couple more years, maybe. It might be gone. Nels, do you know Jim previously as well?
Well, I don't know, actually, Jim, if we've met in person, but I know Jim by reputation and through colleagues and science buddies. So it's really fun to have this chance to kind of sit down and talk.
Jim, let's get a little of your history. Where are you from originally?
I grew up in Wisconsin. I went to school as an undergrad at Texas Tech University. Don't ask why. The reasons weren't very good. It did get me out of the winters in Wisconsin.
Now we're talking.
I went from there to the University of Utah, where Nels is now.
Yeah. So I have to ask, Jim, when were you here and how long?
71 to 77, although I spent 18 months in Australia during that time.
Oh, wow.
And the valley there wasn't quite as filled in as it is now.
Yeah, things have changed a lot. That's for sure. But Vincent, I think this brings up an important side point I'd like to make, which is all interesting biology somehow or another feeds through Utah.
I guess I don't do anything interesting then because I've never been there.
No.
Well, I've visited, but I never worked there.
Yeah. So Jim, was John Seeger or Fred Adler or any folks like that around at all?
I don't think Fred was. John Seeger hadn't been hired yet. Mario Capecchi was hired while I was there, probably in my first year or so.
Oh, cool. Yeah, so Mario is still going strong. His lab is just one floor beneath mine here in the Department of Human Genetics in the medical school. And so we cross paths quite a bit. And so these fellows, John and Fred, are great, I would say, mathematical biologists. John, a great population geneticist, evolutionary geneticist. And I've had some great opportunities to interact with these guys and their students. So one of Fred's students, Laura Struby is her name, is doing some really cool modeling on some kinase activity with a family of kinases that respond to stress and control protein translation. And she's doing all these really kind of sophisticated differential equations to try to get at some of that biology. It's really been eye-opening for me, a whole new sort of part of research that I haven't been familiar with. And so it's kind of fun to think about some of these other connections.
Jim, when you went to Utah, was that for a PhD?
Yes. And it may surprise you that I actually worked with someone who studied turtles. So I wasn't much of an evolutionary biologist at that point in time, but partway through that degree program, I changed fundamentally what I do.
Mm-hmm. And then you went somewhere else to do a postdoc, is that correct?
Went to Madison, Wisconsin with Jim Crow, and then to Sussex, England with Maynard Smith, Brian Charlesworth, and Paul Harvey.
Couldn't stay away from Wisconsin, huh?
I spent a lot of time outdoors as a teenager, and when you have a week that never gets above 0 degrees Fahrenheit outside, you kind of wish you were in a warmer place.
Yeah, I'm sure.
Yeah. I grew up in Minnesota next door, so I can relate to that. And I would say, I mean, regardless of where Jim Crow is, I think you would follow him. I met him a while ago. He was one of the giants and an amazing scientist in person.
Yes, he was. And he was incredibly generous. He was a wonderful person to be around.
How long did you work with him?
I was there a couple of years. I actually went to England part of the time and then came back. So I was back and forth between England and Madison.
What were you doing in England?
Working. That was mostly doing theoretical work with the crowd at University of Sussex. And then in Madison, I was doing some theory work, but I was also working on sex determination in turtles, which at that time we were realizing is controlled by incubation temperature of the egg.
And was Texas Austin your first faculty position?
Yes, my only one.
Yeah, so you've been there a while. I noticed on your, I guess it's your Skype thing, your birthday is an incredible date.
I don't reveal my birthday, so I put down January 1 just to...
Oh, that's fake.
It's fake.
Oh, because I'm January 2, 3 years later, but that's all fake, so...
My mother was January 2.
Yeah, I'm January 253. So I was looking at that and I said, wow, not only is he close to me, but that's quite a day in history. 1950, January 1, second half of the century, but it's all fake, so the hell with it. Okay. Well, Jim, Nels suggested we do this interesting paper, or talk about this anyway, that you have got, I guess, in review at Genes, Genomes, and Genetics, right? Called Lethal Gene Drive Selects Inbreeding.
Yes.
And this is something you put up on Bioarchive as well, right?
That's the first time I've used Bioarchive or any of those preprint servers. But yes, it's up, and it's getting more attention than the papers I published by the normal route.
Yeah.
That's interesting. We've been talking a little bit about the Bioarchive, and I'm curious to kind of get your impressions as a first-time user. I'm a first-time user myself pretty recently. I'm
stunned at how much attention it's gotten. Colleagues have said, oh, I saw your paper up there. I was on the phone with a writer for the New York Times today because of that paper, and I think it's amazing. I'm going to keep doing it.
Yeah. And is this part of this new deal where you can, so when you submitted it to G3, that you did it through the Bioarchive, or did you do that independently?
G3 said, do you want us to put this on Bioarchive? And I said, sure. And I did that. And the paper was rejected. Nothing was wrong with it in terms of the content, but they wanted me to do a lot more. And so when I resubmitted it, I had to upload the revision to Bioarchive myself.
Gotcha.
First time around, the journal did it for me.
Okay. Yeah, I think more, it looks to me, and I just saw a tweet about this from ASM, American Society for Microbiology, that all of the journals sort of under that umbrella, what you can do is go in, if you're going to submit a manuscript to them, you can actually do it through the Bioarchive. And so, I mean, I'm pretty intrigued by this because it's sort of, as you're just sort of bracing for the whole editorial and review process, you have this opportunity to get it out there to colleagues and the science-curious population. And as you're mentioning, Jim, I mean, it sounds like it's been a great way of kind of getting your really cool ideas in front of a broader audience immediately.
I think that was the purpose of it, and I'd say it's working. And, that's great, right? We're concerned quite a bit with getting, if we have an idea, then we're the first one to have it. It's nice to get it out there rather than have to wait 6 months for a review process and a revision process. So I think this is a great thing.
I think that's the key, that why do we have to wait 6 months to a year to see work that's essentially done? We're just waiting for journals and reviewers and revisions and copy editing and all this crap. I think it should be out there. I love it. And about 2 months ago, I wrote a blog post on an article that was on Bioarchive. I got such pushback from so many people saying, you can't write about that. It's not peer-reviewed. As if peer review, is the
yes, right.
what makes science great.
As we found out now many times, you can get anything published supposedly with peer review, and in many cases not, if you choose the right online journal.
That's right.
Yeah.
That's right.
Yeah,
Well,
yeah.
A number of individuals came to my defense and said, look, you peer reviewed it. You read it. You decided it was worth writing about, and that's good enough for us. I said, thank you. That's the way I viewed it.
Yeah, and it's been great for our podcast. I mean, so this episode and the one before, we kind of dipped into the Bioarchive, and I think this has really, helped us to facilitate what we're trying to do here in terms of getting some of the cool work out.
Now, I would guess, Jim, that this New York Times reporter is Carl Zimmer because he has a big interest in evolution. And I talked to him on one of my podcasts a couple months ago, and he said he hates this whole idea of embargo, this artificial release of a paper on an X day, so you get a big press splash. So I'm sure that he and other writers are going to like Bioarchive a lot.
Yeah, interesting. The reporter was Amy Harmon.
Oh, okay.
I agree that Carl Zimmer would have been the logical person, but this was someone else.
Interesting.
Yeah, Amy's great. I met her a couple of years ago, and I think the work she's doing is really cool. So I think, wow, very good.
But it's interesting, I'm curious to see how the press deals with Bioarchive, right? Because they're not all qualified to say whether the experiments are right or not, or whether the data support the conclusions, for the most part. And I'm just wondering how they're going to deal with that, if they're going to get people to help them, or if they're going to wait, or what.
Well, and I think this is actually signals a bigger challenge to our entire community, which is as more and more folks start to, in essence, flood the Bioarchive with work that is at various stages of sort of quote-unquote readiness to kind of be released into the wild. How do we sort of filter signal from noise in new ways? Because if you have, currently I think it's early enough that people are kind of trolling through and finding stuff. But if you sort of multiply that by orders of magnitude of submissions, then you sort of create a new problem, which relates to, I think, what you're saying, Vincent, which is how do we judge sort of quality from quantity in some of these cases.
I would like to comment, though, that so I teach a class for non-majors, and it's really about the interface between science and society, and I'm collecting lots of material, and there are more and more cases of published studies that are bogus, or I guess they're what called p-value sorting. You can do a study, you look at a lot of different things, you'll find some that are significant by chance, they get published, especially if the industry supporting the study finds them in their favor, and the peer-reviewed publications are being flooded by that where there are commercial interest. And so I don't know that Bioarchive, in some ways, Bioarchive might be less prone to these biases in the published literature. But there are more and more cases, especially with nutrition and food, where we have completely nonsensical articles published that had statistical significance. And so this is everyone's problem.
Yeah, point taken.
Yep.
Absolute. Well, why don't we dive into the work, actually? So the Bioarchive paper that's under review currently in G3 is called Lethal Gene Drive Selects for Inbreeding. And so maybe we should just start with some definitions and ground rules here. So what are we talking about in terms of gene drive? What's the concept here or the idea?
So there's a whole range of possible engineerings that might fall under the category of gene drive. The general principle, depending on your definition, is that you can force genes into a population by any of various mechanisms and get them to sweep through or go to high frequency, maybe to fixation. The most extreme forms of that principle would be that you can introduce, in principle, as little as one individual into a population that carries a specific type of gene, and that gene will, because of intrinsic forces of selection and the way it's engineered, that gene will force itself through the entire population of that species. And so we've known about, so this is a category of what we call selfish genes. And when we use that term, we mean it's a gene that benefits itself without necessarily benefiting the individual that carries it. And in extreme cases, selfish genes can cause, while they're spreading, they spread to fixation, and in that process, at that endpoint, they actually cause the population to go extinct. That seems to be a contradiction in terms. We, as evolutionary biologists, we use Darwin and survival, the survival of the fittest metaphor, etc. But it's been known since the 50s that these types of things do exist in nature in some cases. The mathematics of simple cases were worked out over half a century ago. And what's changed now is we can actually engineer many of them. So we can try them out de novo, a priori, and see how well they behave according to our models.
Yeah, and so I think that's a great point that you raised early on, that we're sort of in this new era of gene editing, and in particular, all this excitement about CRISPR. Do you remember, Vincent, have we spoken about CRISPR much on our podcast yet?
I don't think beyond mentioning it, we really have gone into it, no.
Yeah, and so this is this really cool new system that sort of emerged just in the last few years. It's kind of frightening how fast this has moved into molecular biology and genetics. But it's basically harnessing the adaptive immune system of bacteria, where bacteria evolve.
And archaea.
And archaea, thank you. Yeah, good point. And who knows where else we'll find it next, probably, right? But so these critters that are infected by bacteriophages, in the case of bacteria, and related viruses, a way of blocking them through chopping, targeted chopping of DNA. And so this has been harnessed by some folks in Berkeley, in Boston and other places, and has sort of mushroomed into this massive area of new possibilities of genome editing. And so that, I think, opens the door to this, like, as you mentioned, Jim, that these ideas have been around for 50 years, but to actually sort of grasp the ability to apply them now that we have these new gene editing tools, it really sort of comes to the forefront.
And there was a paper by Austin Burt in 2003 where he actually proposed this. He didn't propose CRISPR, but he suggested that we're going to be able to engineer these things, and he talked about some of the problems that we're likely to face, but that they could potentially be used to cause extinction of species. And I thought it was nonsense when he proposed it. I thought we were never going to see the day when we'd be able to engineer these things, and like 10 years later, and it's here.
So you call this a homing endonuclease gene in your paper, and maybe you could explain how that, in principle, how it would work using the CRISPR-Cas system.
So the key to many of these gene drive systems is if they're going to cause individuals to die, they have to have some advantage to offset that so that they can spread. And the key to many of these gene drive systems is that when they're in a heterozygote is an individual in which one of its chromosomes carries this homing endonuclease gene or this selfish gene, the other one doesn't, that selfish gene can double itself. It can take over the other chromosome. So you go from an individual carrying one copy of the gene to 2 copies of the gene. And all it needs to do is do that in the germline so that the gametes transmit that way.
And maybe we could even just define homing endonuclease. So what is the selfish activity here? How does this work?
So in the CRISPR model, it works as follows. So CRISPR is Cas9 is a protein, and it uses a guide RNA to decide where it's going to cut. And I don't know a lot of the details about this, but it has a specific set of sequences, and when it recognizes those sequences anywhere in the genome, it will cut that. Mm-hmm. It's a nuclease, so it'll cut both strands of the DNA. And that's really all it does. And so those broken DNA strands are sitting there. Well, the cell has machinery that looks for that. It sees the broken ends and decides, oh, this needs to be repaired. So it starts looking now for homology. It's looking for a template to copy that to repair that sequence, because it doesn't know that the DNA has just been cut. For all it knows, a whole big chunk of DNA has been taken out. It's just got loose ends. And so it looks back to the template strand that isn't cut, and and so if the CRISPR gene is sitting on the template strand where this other chromosome's been cut, then the cell copies CRISPR into the cut part, because it doesn't know that CRISPR doesn't belong there. So you go from so CRISPR needs to cut opposite where it is in the chromosome. So it needs to take a chromosome that doesn't have it, but cut at the spot where it sits in the chromosome where it does exist, which means, so really all it's doing is cutting at a particular site. The cell does the rest. The cell looks back to the uncut strand and says, oh, here's what I'm supposed to copy. And now it copies in CRISPR where it didn't exist before. I don't know if that was clear.
Yeah. And so the outcome of that, I guess, is that if that is targeted to an essential gene of the host, so to speak, then that will cause a detrimental effect in terms of fertility or viability. And then that's the basis sort of this selfish activity, the spread of the drive system at the cost of the organism being able to continue on. Is that right?
That is correct. But what we need is that as long as an organism has one functional copy of the gene, as long as the individual has one functional copy of the gene, it's mostly okay. Mm-hmm. If CRISPR happens to sit in a gene where the organism needs two copies, that isn't going to work because the organism's dead. Mm-hmm. So you want a situation where as long as I have one functional copy of the gene, I'm okay. And then CRISPR should only jump in the germline so it's not bothering my somatic tissues. That is, I can still be a functional individual. In my germline, all the chromosomes are getting converted to convert CRISPR. They're destroying this essential gene, but as long as the essential gene's only needed in my somatic tissues and one copy's enough, everything works out fine. Mm-hmm. And that means if a zygote is formed where CRISPR is inserted into this gene and from both parents that are forming the zygote, that individual's dead. But as long as, if just one of the parental chromosomes carry CRISPR, the other one has a functional copy of the gene, we're okay.
But if you get conversion in somatic tissues, it probably doesn't matter all that much, right? Because you would never convert every copy in all your somatic cells.
I suppose that's a matter for debate, and that would be how good the engineering is too.
Yeah, I guess. I suppose. Yeah.
Yep.
So we talked about this on TWiEVO months ago in terms of releasing mosquitoes with a gene drive cassette like this, to limit parasite or virus transmission. And we had a big discussion about whether you would get mutation to avoid this, and that's something that's obviously the crux of this paper. And it seems to me that if you stick a CRISPR-Cas cassette in a gene, there's no selection to maintain it there. So why doesn't it just get removed?
Because it's its own selection. Mm-hmm. The fact that so let's say you have a mutation that knocks out CRISPR in some fashion. It's no longer functional. The rest of the population is still going to have active CRISPR, and those are going to keep spreading. The CRISPR that got knocked out will sit there up until a point. So it kind of depends on some details here. That CRISPR locus will be, the mutated CRISPR locus will itself be protected from further CRISPR attack because it's no longer got the recognition sequence that CRISPR wants to cut. But the rest of the population, CRISPR is just going mad in that. And furthermore, if CRISPR is mutated so that it is sitting there but non-functional, there's no particular advantage to that. It's going to be lost, right? Because that's a recessive lethal mutation. Now, suppose you had a type of mutation which completely deleted CRISPR and restored the original sequence. CRISPR can invade that again.
Yeah.
Suppose it modified the original sequence a little bit. Well, now that might be, let's say it's modified the sequence a little bit, but the gene has become functional again. That type of mutation will actually spread because it's functional, CRISPR can't invade it, and so if you do the mathematics of that, you'll find that that starts spreading as CRISPR becomes more common.
So it also seems like people have thought about some of these kind of workarounds where you might have some point mutations or some compensatory mutations that would somehow disrupt this, in which case you might, go back with multiple CRISPRs at once or multiple homing endonucleases at once in order to try to do that. But I think what's really cool about the work that you're considering is to sort of step back and think about, well, what are other mechanisms beyond just sort of that sort of narrow view of what might happen just with the selfish genetic element, but instead what might happen with the whole population. And that moves us, I guess, to this idea of inbreeding showing up or selfing showing up as another evolutionary mechanism of resistance to a gene drive scenario.
That was, in fact, the point of the article. And there are precedents for that. I think Austin Burt in his 2003 paper did suggest that, if the species was already inbred, then that's going to work against this homing endonuclease gene spread. The question is, if you start with an outbreeding population, will an allele that sort of increases the level of inbreeding spread in response to homing endonuclease gene? And that's much less obvious because let's say CRISPR or your homing endonuclease gene is out there and you start inbreeding. Well, one of the things it's going to do is it's going to make you more often homozygous for this recessive lethal gene. So inbreeding, it's not clear which way selection is going to go, even though it would be good if you were highly inbred and you could purge this CRISPR gene. It's not clear that you can get there by evolution. Mm-hmm. And it turns out that when you do the math, you can actually get there.
So, and what are, thinking about what are the kind of gene drive examples or species that has this been tried actually in the wild or in the laboratory? What are the sort of
so someone did engineer a homing endonuclease gene in Drosophila. It was published in Science a year or so ago, and it did work as expected, at least in broad outline. I don't know that they looked at whether it was a 99% gene conversion in the heterozygote or not, but it did work as somewhat as expected, but the first realizations of this type of selfish gene, a gene that has an advantage in meiosis and that it goes to most of the gametes instead of half the gametes, that there were a couple systems discovered in the 50s, natural systems, one in fruit flies, Drosophila, and one in house mice, the T locus system in house mice and something called segregation distorter in Drosophila. And geneticists jumped on those right away because, we'd had half a century of Mendel, and suddenly we found systems that weren't behaving as Mendel. And the mathematics of some of the simple cases were actually worked out in the 50s. But we couldn't really, and people did have the idea that these selfish genes might be used for some kind of population control, but if you can't engineer them, you're really limited in what you can do.
I don't quite get this, maybe because I never was a good genetics student, but can you explain why inbreeding would solve this problem as opposed to outbreeding?
So the homing endonuclease gene depends on this advantage of being in heterozygotes. Mm-hmm. So mom gave me one copy of it, dad didn't, or vice versa.
Right.
And now the homing endonuclease gene can jump across. And this advantage in heterozygotes is what drives. It gives the gene its advantage despite its tremendous disadvantage in being a recessive lethal. If you go, let's say, take the extreme where everyone's inbred, right? So you have no heterozygotes anymore.
Mm-hmm.
So everyone's either homozygous for the homing endonuclease gene, in which case they're dead, or homozygous for its absence. And so you can see in that case, it's almost impossible for the homing endonuclease gene to spread at all.
So when you do inbreeding, you can get some wild-type homozygotes that would not have the gene.
That's correct,
right?
Yes.
Got it. Now, I know about inbreeding in plants, but what about in other organisms? How does that work?
So my model was restricted to plants or hermaphrodites in general. And most of the organisms we're concerned with aren't hermaphrodites. They have males and females. You can get inbreeding there with sibmating, with certain types of group structure. It's not clear. I think broadly the results I have for hermaphrodites will carry over, but there may be many details that differ. In general, it's harder to get high levels of inbreeding or inbreeding coefficients when you have separate sexes than it is when you're selfing.
Mm-hmm.
But there are plenty of ways that you can have inbreeding with separate males and females.
Mm-hmm. So, and it seems like a lot of the excitement and movement towards testing out these gene drive systems is focused on insects now, and especially the vectors, the mosquitoes, right, that carry these viruses. And so I was kind of curious, are there hermaphroditic insects out there actually, or is this all?
I should know the answer. I don't know of any. So, but again, this is a highly artificial model, and I'm guessing that the results will carry over to other forms of inbreeding.
Mm-hmm, mm-hmm. Yeah, so I was kind of curious. I actually did an internet search of this, and this will be my pick of the week later in the episode, is I guess there are some very rare cases of hermaphroditic insects. And that got me thinking, I guess this might be a little bit wild, but that if enough gene drive is done, whether this would put some selective pressure not only for inbreeding and sort of sibmatings and so forth, but if this could even start to foment possibly hermaphroditic systems to emerge, since these would obviously have an advantage if the gene drive was sort of pervasive and strong enough. But I think that might be, I don't know how rare these events are, probably super rare.
Yeah, I think that would be a big challenge. I think it's going to be much easier for things just to evolve inbreeding the old-fashioned way without going through hermaphroditism.
Yeah, yep. So, and actually so part of when you're setting up the model, I'm just curious about why you chose to use the hermaphroditic or selfing as opposed to inbreeding. I guess it's just less complicated for the modeling?
Yeah, fewer equations to write and simple equations.
Mm-hmm. So can you give us an overview of the model and how you programmed it and what happens? It
assumes two loci. One locus has the homing endonuclease gene, and there you only have the wild-type homozygote and the heterozygotes for the homing endonuclease gene. The homozygotes for the homing endonuclease are dead. And the other locus has got two alleles, and I assume the population was initially outcrossing, so the new allele at that locus increased the level of selfing. Mm-hmm. And you write out some equations for what's going to happen in the population. I did most of it by simulation, but some of it was analytical. But you run through lots of cases because you have to ask, will an allele that increases selfing just a little bit be favored, or do you need a big jump in inbreeding? And there are many cases where you actually need a big jump in inbreeding for it to be favored at all. But you run through a lot of cases on the computer, and there were some anomalies. There were cases where, so you'd think that homing endonuclease gene with this profound advantage, where I assume, by the way, that the heterozygote was always converted completely 100%. You'd think something with that advantage could never be kicked out of the population, but there were actually trials, depending on the starting frequency, where the homing endonuclease gene came in, selfing was favored afterward, and then you get into these cycles where suddenly selfing drives the homing endonuclease gene frequency down, and it actually disappears once it's gone. And I set up a case where selfing wasn't intrinsically favored. And so in that case, selfing was later lost. So you had this paradoxical outcome that you ended up losing both the homing endonuclease gene and the selfing. But those cases weren't very common. They just indicate that the system's going to be pretty complex to study in any real case.
Yeah, and so to kind of cut through some of that complexity, I mean, what are some of the key parameters here? So you mentioned you have these sort of two dual allele scenarios, the drive, the endonuclease, and then the selfing. But you also mentioned, so male drive versus two-sex drive is another parameter here. Could you explain that a bit?
So when you engineer these, first of all, when you find these in nature, the drive almost always operates just in one sex. Could be male, could be female. The first few cases, the T-locus system in mice and segregation and distortion in males, both of them were in males, in Drosophila mice. If you engineer these to jump, the homing endonuclease to jump, right? You don't want to turn this on in all the cells for the reason we talked about, that you don't want your somatic tissues to become converted to be homozygous for recessive lethal. So you want these to be turned on just in the germline. And so you can do this, but you'll have to come up with different constructs to do this. It's going to be easier to do it in just one sex. And so if you have a homing endonuclease gene that's moving only in one sex, you do not get the same level of population suppression that you do if it's driving in both sexes.
And actually, are most of the systems that are being sort of imagined or envisioned, are these two-sex drive systems for gene drive in order to get that? Or what are the maybe trade-offs with that? Like you're saying, maybe you lose these.
I'm not actually that up on what's being planned. My guess is it's going to be, if you do two-sex drive, which the predictions are you'll get much better control. If you do that, that's going to increase the different ways that resistance can evolve. And and I think, the outcomes of these systems are highly sensitive to the rates of gene conversion and so on. And I think it's going to be harder to engineer systems where you have really tight control over everything in both sexes. Yeah. So there's going to be a trade-off there.
Yeah. Kind of you're damned if you do and you're damned if you don't with evolutionary resistance cropping up. Yeah. So and then there's
another parameter I'll talk about. But yes, if a gene drive is engineered just in one sex, you only knock out half the zygotes when the gene drive has gone to fixation. And half might sound like a lot, but depending on the ecology of the species, and it'll be mostly zygotes that are dying, and that may have relatively little impact on the number of adults that survive.
So, and then the next, I think, key parameter that maybe you're getting to is this idea that as the drive sort of ensues or continues on, that then sperm sort of has a different value, is the way you describe it. Either sperm is discounted or valued in a sense.
Okay.
What's that parameter?
First of all, let's deal with that second. Probably the most critical parameter in all of this in terms of what impact selfing or inbreeding has on stopping the homing endonuclease gene is something called inbreeding depression. It's how much an inbred offspring is intrinsically lowered in fitness because it's inbred. So most of us understand the concept of inbreeding depression. That is, when you inbreed, offspring viability, fertility goes down. It varies a lot between species and how severe that is. Some species it's very severe, other species it's not so severe. Across the different models I considered, inbreeding depression had the biggest effect on predicting what the outcome was in terms of how effective inbreeding was at stopping a gene drive system.
Mm-hmm. So this is the kind of the balancing act here, I guess, where the gene drive is having a negative impact on the population, but then inbreeding itself or selfing can have negative impacts. And so at what point do you sort of tip the scales one way or another?
Very good. That's a very good way of describing it.
Yeah.
So then that brings up your next point. There are different ways of modeling selfing. And this may be specific, well, I guess it would apply to other forms of inbreeding. Let's think of a plant, but it works for males and females too. If to self offspring, you use up almost none of your pollen, then that's a case in which you can produce, you give up some of your seeds to selfing, the others remain outcrossing, but your pollen pool, the pool you're contributing out there to fertilize seeds on other plants is almost not affected. Mm-hmm. Alternatively, it could be the case that, well, to self-offspring, you have to, let's say you're going to self half your offspring. Maybe you give up half your pollen. Maybe half your pollen is lost to those self-offspring. And so now what you contribute to the pool of pollen that goes out to other plants is in fact half what it was before, half what it was if you didn't self at all. Those are two extremes of a continuum that people have recognized over the years, and those two versions make a difference to how effective selfing is. And you might intuitively guess, and it would be correct, that if your pollen pool isn't very much affected by how many of your ova you self-fertilize, then selfing is even more effective at stopping a gene drive system than if your pollen is discounted by how much you self.
Interesting. So then I guess the parameters you considered, the male drive versus the two-sex drive, and then also the amount of inbreeding depression, I guess that those led to pretty different outcomes depending on how those variables.
I would say they led to quantitatively different outcomes. So the system that is most sensitive to the parameter values in terms of whether selfing will be favored at all in response to a gene drive system is a one-sex drive case. And you'd expect that because the most you're knocking down population fitness is 50%. And so if inbreeding depression is too extreme, if, let's say, inbreeding depression is more than 50%, that doesn't work well, at least in some cases. There are lots of things that can get in the way of selfing stopping a gene drive system because the penalty for the population of the gene drive system isn't that extreme. It goes back to your point that you're balancing several things here. In the case that the gene drive is operating in both sexes, that thing, if it's left unfettered, untouched, will drive the population extinct. It knocks mean fitness down to zero. So that leaves you a lot more room for even high values of inbreeding depression to get in the way.
I see. So maybe could you take us behind the curtain a little bit about actually how you do the computational modeling? So like you kind of hinted at that you're doing simulations here, but I'm curious sort of how the runs are done or how much even computational horsepower this takes. And
almost none. Well,
yeah.
I suppose it depends in some ways on how extensive the simulations are, but you write down some equations, and there are, I don't know, only about 6 equations for some of these things, maybe a few more. But I just wrote a program in a standard programming language. Recursion equations are actually pretty easy to deal with. There are no graphics involved in the runs, whatnot, and you just iterate these, you run these through thousands, tens, or hundreds of thousands of supposed generations, and you keep spitting numbers out. The numbers represent what the frequencies are of the different alleles in the population. And it's, I don't know, I consider it pretty straightforward, but the key isn't the simulation so much as it's writing down the equations in the first place, which takes pencil and paper.
Yeah, and that, I guess, sounds like the heavy lifting here is to sort of take the complexity of the biological reality and the many variables and sort of simplify that or try to capture that in a set of equations. So
that is almost always the trick
yeah.
in modeling is how do you capture the physical or biological reality?
Uh-huh. And so do you think that part of that feedback comes from the kind of the output? You hinted at this before, or talked about this a little bit before, that sort of the influence, these paradoxical things that sort of fall out of this, so the influence of the initial conditions or how the drive system might become extinct depending on things like that. How does that sort of play into your thinking as you're developing the model or working on it?
Well, that didn't play into my thinking at all. That's a case where you just run different initial conditions and suddenly you discover that, oh, the gene drive got lost here and inbreeding got lost. How can that possibly happen? But in general, when you do models like this that have many dimensions in them, you find that there are what we would call multiple equilibria, multiple endpoints you can get to that are the system will stay there, right? The gene frequencies will stay there. And so if you have multiple equilibria, then you're always going to have zones of attraction for those equilibria if they're stable. And so then it's just a matter of figuring out, okay, what's the zone of attraction for this? It's really, it boils down to what are the initial conditions that lead to one outcome versus the other. But I didn't expect this to happen at all. I just stumbled on it. And I honestly don't even know if some of these anomalies might be due just to rounding point error, right? Because, if you're driving a frequency down to 10 to the minus 50, and your computer, the software only says, well, once you get below 10 to the minus 15th or something like that, it's, they kind of punt at that point. It may be that some of these anomalies I'm seeing are due to limitations of the computation. But the other side of it is if in a model a gene frequency gets to 1 in 10 to the 15th, right, that's a pretty small number. That's effectively extinct in any real world population.
Yeah, absolutely.
How many generations does it take to lose the endonuclease?
You mean in the runs where it's lost?
Yeah.
So actually, it was about. Well, it depends on the starting conditions, but from the starting conditions, things happen very fast, even though you're starting at moderately low frequency for some of these things. Things happen very fast, and in the course of 30 generations, your homing endonuclease gene has shot up to high frequency, your selfing allele has shot up to high frequency. By 30 generations now, the homing endonuclease is down to be relatively uncommon in the population, but then you sit, there's another, I guess it depends on what you want the end point to be, but in the course of the next 1,000 generations, you've effectively lost the homing endonuclease gene, and now you're in the process of selecting against the inbreeding allele because the only consequence of that is inbreeding depression.
Yeah, and this sort of rapid stuff. So we've talked a little bit in a previous episode about segregation distortion. We had a Drosophila speciation geneticist, Nitin Fudness, who's a colleague of mine here at Utah now, just discussing this. And it seems like in these cases, things can move really quickly. And I'm thinking about, there's this really great book, Genes in Conflict by Austin Burt, who you mentioned earlier, and Bob Trivers. And,
yes.
Yeah, and some of these kind of wild natural cases. There's one I remember, I think a segregation distorter where a population of clams all of a sudden becomes like 90% plus male, and this sort of huge imbalance in the male-female ratios, and then an ensuing crash of the population. So it seems like, would you say you're modeling here of the gene drive where where basically folks are purposefully trying to really drive a hard strong selection, that this sort of matches even some of these kind of natural scenarios, whether it's the T locus or the other ones you mentioned?
Yes, I would. In a general sense, they do. I think what we're going to find is the success of these interventions we're engineering is, we're starting with a general concept of segregation distortion. The details are probably going to matter. And so I expect some of these things may work very well and others may not. And it's going to be hard to predict until we actually do it. One mosquito may succumb to it and another one not. And then when we look at why, it's going to be, some nuance of the species-specific biology.
Mm-hmm, mm-hmm. Vincent, do you know this book, Genes in Conflict?
I don't. Should I read it?
Yeah, I'll get you a copy. I think it's worth taking a peek at. Yeah, it's really, this is kind of one of these formative books. It certainly was for me and others of kind of my mentors and friends. Do you know that you know this book, Jim?
Yes.
Yeah.
So people are in the process of designing gene drive scenarios in mosquitoes for eventual release. I don't know the details, as you said earlier, Jim. You don't know either. But at some point, one could, in the lab, look at some of these scenarios, right, Jim? They could mate mosquitoes that they've produced and see what happens. Isn't that correct?
They almost certainly will want to do that in the lab first, because if you engineer something that isn't quite the way you need it, and you release it, you might actually interfere with future attempts to perfect it. Mm-hmm. And of course, there are people legitimately worried about these things jumping into other species. A friend of mine, Harmeet Malik, who's at the Hutch in Seattle, said, what if one of these jumps into monarch butterflies and we lose them? I think these are big concerns, and you can say, well, how is a mosquito gene ever going to make it into, say, a butterfly? But there are these transposable elements called mariner, and they move around across the huge taxonomic jumps, and we have no idea how they're doing it. So it's not out of the question some of these things could jump around, but then you have to balance it by, look, suppose we really could knock out Aedes aegypti. I mean, the impact on world health would be absolutely huge. So we got several things to balance here.
Well, there could also be negatives of knocking out Aegypti, right?
I will probably dodge that one to some extent. I will point out, however, that Aedes aegypti is now found many places it wasn't historically.
Yeah.
And so it's been an invading species, and I think it's much harder to make the case there'd be a negative ecological impact.
Yeah, I think you're right.
Us to the former state.
I mean, I was just reading today about Aedes, and the current species that bite humans are actually, they've evolved to be peridomestic, and they don't even like wild animals anymore. They prefer us, whereas the Aedes species that bite animals exclusively are elsewhere. So, and they're separate species, so maybe you could design this so that it would not affect the animal-preferring species, right? Because that's the concern, that you interfere with an animal cycle and throw things.
Yes, yes.
Yeah, and it also seems like, there have been some attempts at biological control using at least related things. And you bring this up, Jim, in the paper, sort of these introducing sterilized populations that have been irradiated or so forth. I'm also thinking of, the release of myxoma virus to rabbit populations in Australia as a way of doing biological control. And it seems like in all these cases, resistance does show up, or it sort of fades and these populations rebound in different ways. Mm-hmm. But I guess the question then becomes whether what we're on the cusp of here with gene drive is just sort of fundamentally different in its power. But I think, at least for me, one of the take-home messages here is that they're at these various layers of kind of biological organization where you could start to see resistance, whether it's, a single change that removes a endonuclease cut site in a genome, all the way out to these differences in who's mating with who, which is really important to think about this whole playing field of possible resistance mechanisms.
It's also worth acknowledging that in the sterile insect technique, they've tried this on. So the sterile insect technique is usually known as irradiating males and releasing them so that when you're in the lab, you have big populations of these things, you irradiate them, you release them in the wild so that when they mate with wild females, all the offspring are dead because the chromosomes are broken up by the radiation. that technique's been applied around the world to multiple species. There's successes, there are failures. One of the big successes is what a fly called the screwworm, which used to get up into the plains in the U.S. In the summers, up into the 50s and early 60s. It was the scourge of the cattle industry, because this fly would lay eggs in any kind of open wound, and the eggs would hatch, the maggots would start devouring the live flesh, make the wound worse, and they could kill a cow in something like a week. The timing of the birthing of cows is still sort of oriented around that. They tried to do it in the spring before this fly had time to spread. It would overwinter in southern California, southern Texas, and southern Florida, and then the population would expand over the course of the summer. So they wanted the birthing to happen before this fly actually got to many of the locations. Sterile insect technique is used successfully to knock this thing back to Panama. It's now in South America, but it's been an extremely successful program, despite the fact that it hasn't worked in all species that they've tried it against. So there, I think, with the gene drive systems, we're going to be looking at the same thing. There are going to be some successes, there are going to be some failures, and we'll learn a lot from it.
Yeah, wow, really cool. So, Vincent, this is our first computational modeling paper.
Probably won't be our last either.
It won't be our last. It was good for us, I think, to dive in and try to sort through a few equations here and there. But I wanted to ask Jim, I know we don't want to keep you too much longer, but kind of how you balance. So one of our themes on TWiEVO so far is balancing or sort of integrating both computational evolutionary approaches as well as experimental approaches, which seems like it's something you do really gracefully in your lab. And I'm thinking about all of the experimental evolution you guys have done over the years with viruses. Both Vincent and I saw your recent PLOS pathogens review with one of our science buddies, Adam Loring, up at University of Michigan on both modeling and empiricism and virulence evolution, thinking about viruses again. But maybe say, if you would be willing to say a little bit about that, how you kind of think about both the computational and experimental approaches in your work.
I use whatever is convenient, and empirical work is hard. And over my career, I've oscillated between emphasizing theory more than empirical work, and vice versa. The one thing I'll say is it's very difficult to do theory and get the predictions right, at least in a quantitative sense. It's really easy to be wrong. It's not that the model is wrong in the narrow sense of the word. It's that you set the model up a certain way, you expect certain outcomes, and then when you try and do the biology, it's like the biology hasn't been reading your paper and goes off in a completely different direction than you expected. And so that's the big challenge, is it's really easy to think your theory is right because you work on the mathematics so long and you kind of expect that the model, nature will behave a certain way, and some assumption that you never really thought about turns out to be critical, and is why when you do the empirical work, it doesn't turn out the way you think.
But it seems like some good feedback then from kind of the conversation almost between the theory and the experimental results to then
that's exactly the way it should be done.
Yeah.
I think this is a key paper to come out now, because right now everyone's talking about wiping out mosquitoes because of Zika particularly. And I think we need to have papers such as this one to say, hey, it may not be as straightforward as you think. And I'm sure that's in part why the journalist was interested in this, I would guess. Mm-hmm. Mm-hmm. So, Jim, you, we're going to let you go now, but I was wondering if we could get you back in the future, because I'd really like to talk about the evolution of virulence. Would that be something you'd be interested in?
Sure. I'd be happy to do it. I've enjoyed this a lot.
Because I think about that a lot. I read your review with Adam Loring during a train trip, and actually I was emailing him back and forth, asking him questions about it. It's something I think about all the time. So, I don't know, Nels, would you be interested in that?
Yeah, that would be really fun.
Yeah, so we'll give you, some time to recover, but
we need some too.
I think TWiEVO would be the right place for that, right, Nels?
Oh, I agree. And this is something, I mean, that's kind of near and dear to our hearts as thinking about viruses and how they evolve. And virulence evolution, I think, is just like a fascinating topic that hits, again, all of these themes kind of all across the board. And so that, it would be really fun to revisit this soon. Yeah.
Very good. Yep. Jim, we'll let you go. Thank you so much for joining us.
Thank you.
This was great, Jim. I really appreciate it. That was fun. This was fun.
Well, it's good to talk to both of you.
All right. Catch you next time. Bye-bye.
Bye.
Jim Bull, University of Texas, Austin. Cool stuff.
Yeah.
Cool stuff, Nels. Nice job.
Thanks. I really enjoyed this. I actually think it was your idea to do this Bioarchive paper, if memory serves correctly. So
I don't remember. You can take the credit.
Well, okay. Well, how about we share the credit?
That's a good idea. Before we do something else here, we'll do a couple of emails. You have time, right?
Yeah, absolutely. That sounds good.
I want to tell everyone about our sponsor for this episode, CuriosityStream. It's the world's first ad-free nonfiction streaming service. They have over 1,400 titles and 600 hours of content, founded by John Hendricks from Discovery Communications. So you're guaranteed access to real science shows, not reality TV shows currently plaguing cable TV. It's available on many platforms like the web, Roku, Android, iOS, Chromecast, Amazon Fire, and Kindle, Apple TV, 196 countries worldwide. And what they have is a wide variety of science and technology content in addition to nature, history, and many other topics, but it's all nonfiction. If you like truth, if you like facts and investigation, this is for you. They also have interviews and lectures. Their library includes Stephen Hawking's universe. They have a variety of shows on viruses and evolution. They have Life on Us, a series that explores the biodiversity of our bodies, and Secret Life Underground, the secret world of organisms that populate the underground world. So this is why I took this ad for CuriosityStream. I figured our listeners would be interested in all of these.
Yeah, this is making me curious about CuriosityStream.
Well, you can check it out now. So I'm going to tell you right now how you can check it out for 2 months for free. They do have monthly and annual plans available. You can get on at just $2.99 a month, less than a cup of coffee. Just think, forego 1 cup of coffee a month. Of course, for some of us, that's tough.
That's like half a cup of coffee if you're into fancy coffee.
Yeah, fancy coffee. That's true. Or $2.99. You buy a movie on one of their competing platforms. It's more than $2.99. That's right. Enrich your life and continue learning with one of the largest 4K libraries around. I don't know who has a 4K TV. You don't have one, do you, Nels?
No, not yet. And I think we might have even mentioned this before. It's this conundrum of if I buy this, then it's like a 6K TV shows up the next day, and then
Anyway, check out curiositystream.com/microbe. Use the promo code microbe during sign up, and you'll get unlimited access to the world's top documentaries and nonfiction series completely free for the first 60 days. That's 2 entire months free of one of the largest 4K libraries around. Just go to curiositystream.com/microbe and use the offer code microbe at sign up. Make sure you use that, and you'll get your 2 months for free. We thank CuriosityStream for their support of TWiEVO.
Yeah, definitely.
There'll be more advertisers coming, and we have a great audience, so it's a good fit.
I agree. Feels like it's building in a good direction.
You bet.
Yeah.
All right, let's do a couple of emails. A bunch have accumulated now because we've had a series of guests.
Yes, this is great.
Let's do a few of them.
Start to cut through a bit.
You want to take that first one?
Sure, absolutely. Anne writes, Doctors Racaniello and Elde, what an enjoyable educational episode. Dr. Fudness's recounting of the history of the work that preceded his own. So this is Nitin Fudness, who is talking about speciation genetics in Drosophila. Recounting the history of the work that preceded his own and his own work made this podcast an engaging one. I wish there had been a few slides to accompany his talk, as it would have enriched my understanding of the gene interactions, but I'll take a look at the paper. Dr. Fudness's storytelling style reminded me of Eric Lander's way of teaching, as he interweaves the history of science into his lectures. I've taken the excellent MIT X biology course he teaches. Thanks for these fine episodes. Will you be building a library of recommended books and videos for the podcast, as you have done in TWiEV? It would be great to be able to explore some of the books on the topic of evolutionary biology and its interaction with genomics and genetics. Regards, Anne. Thank you, Anne, for this letter. And I will, first of all, pass along some of this info to Dr. Fuddness. I think he would be interested in this comparison to Eric Lander. Convergent evolution there. I don't know. And then also, I think this is a great idea, actually, to start to try to curate some books and other resources. I mean, this is coming up just in the kind of course of our conversations. Today, this book, Genes and Conflict, which I would recommend, and we'll add some links. And I think we should think about this. We're always so busy, but certainly give us a little time, and I think we'll start to curate some resources as we go forward.
Yeah, I'll put Genes and Conflict in the show notes. And
cool.
When you click on it, it'll bring you to our Amazon store, and that will be all the books we've ever talked about on TWiEVO, which is not a lot yet, but there will be in the future.
Yeah, keep building the library.
I think on TWiEVO we do, people pick books a lot or recommend them. I put them on the Amazon store. We have something like 250 books there.
Oh, wow, is that right?
You can just browse it and say, hey, what do I want to read today? Boom. And we get a little bit of, we get a few pennies for each book that you buy there, so we appreciate that.
Yeah.
The next one is from Alex, who writes, Hi, Nels and Vince. Congratulations on another fascinating addition to the This Weekend franchise. It's another beautiful day here in White Salmon, Washington, 16 Celsius and sunny. Mount Hood, brilliant with spring snow. Speaking of snow and ice, in episode 1, Nels mentioned in passing a colleague named Joe Thornton, a biologist and amateur hockey player with the moniker of a very celebrated pro, who was proud of leading his men's league in penalty minutes. As it happens, penalty minutes have been used as a source of data on aggression. Penalty minutes for roughing and fighting are a convenient metric for non-pathologic aggression and are associated with more extreme 2D, 4D finger length ratios, a marker for fetal exposure to androgen, which is presumably the cause of the altered behavior. On hockey teams, there will always be one or two tough guys. There have to be, or the team will be abused by competitors. But a team composed entirely of tough guys is no good either. Instead, coaches balance a team for toughness and skill, but that's all done quite consciously. Evolution here appears to have produced an epigenetic mechanism for modulating individual behavior, but to be useful, that behavior would have to be modulated within the context of other individuals. While it's easy to see how you might get a gene that makes an individual more aggressive, a gene that instead means that individual behavior could be epigenetically tuned is harder to understand. Where should one look for genes which would control the distribution of traits in a population as a coach does in assembling a team, e.g. not the chance that an individual might have a trait or not, but a metagene, one that would control, say, the skewness or kurtosis of the distribution of such traits? Have any such genes been found? Many thanks for another great podcast, Alex. And I don't know, I'm going to let Nels get this, but I'll bet it's going to be an enhancer or something like that.
Yeah, there you go. Right. Yeah, no, this is interesting. So it's kind of fun, and I think a great provocative letter. It's fun to think about sort of how some of these ideas that we think about, like even today, we're talking about these genes in conflict, that book and these examples, these selfish genes, these distorters, and how, even in these simple systems where we're just thinking about how a single gene might move through a population, and then to extrapolate that to hockey penalty minutes feels pretty ambitious to me. But I think it's fun and interesting to do. Getting at sort of the evolution of behavior. I mean, I think maybe we echoed this a little bit in today's episode even. Just doing a simple computational model of how a single gene moves through a population is challenging because you get so many ecological variables that sort of arise. And then, at least for me and my sort of intuition, is that as we then add on kind of social layers and all of the proclivities of human behavior and interactions, whether on an ice rink or anywhere, it becomes pretty thorny or complex. And then when we start to think, like you're saying, is this an enhancer, or the epigenetics, all of the metagenes, all of the contributions, I think there's absolutely something to that. I just think sort of as a, unreformed reductionist or something like that, these are the questions that sort of scare me away in terms of actually digging in and trying to kind of put this into an integrated understanding. But I appreciate the ideas in the letter, Alex. And it is true that Joe Thornton is not only a biologist but was an amateur hockey player, and I think there's a Joe Thornton maybe who plays for the Chicago Blackhawks, which actually is a geographical connection since Joe Thornton's lab is at the University of Chicago. So,
well, the way I look at this, Nels, is we get spoiled by genes that control susceptibility to infectious diseases, say, right? But behavioral patterns are going to be more complicated. It's not going to be just one gene likely that's going to control it. It's going to be a combination or a transcriptional network or something like that.
Yeah, that's right. Yep. And then, when we spoke to, for example, Mike Shapiro, and he does the pigeon morphology and doing the kind of QTL analysis, getting at the quantitative traits or the combination of many genetic contributions, even for simple morphology. Like, so he's gotten some really beautiful traction on getting to some of these simple morphological traits relative to the behaviors. And so there have been a number of people, and continue to be a number of people, who are working on this. Hopi Hoekstra, for example, is in, I think, a really exciting way looking at the behavior of burrowing among rodents, for example. Folks using stickleback fish are thinking about schooling behavior and other really cool behaviors out there. And so far, I think the jury is still out on whether, plenty of QTLs in some cases, but to really nail down kind of a functional connection, we're still kind of in the infancy of doing that. And I think that speaks a little bit to just the complexity of when you start to think about behavior. We just add it feels like almost to me, an order of magnitude more complexity to the questions. And what are you really measuring, and how do you define that in a clean way? That becomes really challenging as well. Like if you get 100 penalty minutes, is that functionally different than 75 penalty minutes or, for an enforcer on a hockey rink? But anyway, really fun to think about, and great. Thank you for the letter, Alex.
I was fortunate to be at a meeting a couple of weeks ago with Hopi Hoekstra, and she was talking about this and how, often science writers don't quite get it, but she mentioned Ed Yong, who nailed it, she said, in terms of understanding that there's not a gene for this or that kind of behavior, but rather it's more complicated. Yeah. And in fact, she turned out to be she's a very nice person. I'd never met her before. And
yeah, she's great.
Invited her on to TWiEVO, and she said she'd be happy to join us. So
yeah, I can't wait. I'm looking forward to that one. Yep. Yeah. And it's a good point you raise, Vincent, about this. I mean, I think historically, even in the last couple decades, there have been just one after another of these kind of reports in the popular press about, this gene or that gene.
Yeah, sure.
The novelty-seeking gene is one I remember from college, actually, which was an allele of a dopamine receptor that was associated with how you answered questions on a survey about would you rather go skydiving or, I don't know, take a nap or read a book or something like that. And, this has been kind of risen and fallen once or twice, and I think currently has sort of been slightly debunked. But this sort of idea, I mean, it's really kind of, alluring to try to simplify this and to, call something a gene for a behavior. But again, that sort of belies the complexity underneath. Yeah.
So just to make it relevant to our conversation with Jim, when I was talking about Aedes aegypti, just today I was looking at this paper out of Leslie Vosshall's lab at Rockefeller, and I'm sure you're aware of this, where Aedes aegypti has evolved to specialize in biting humans, a subspecies of Aedes aegypti. And the reason is because one of the odorant receptors in the mosquito that detects, compounds that are specific to people has changed. Its expression in ligand sensitivity has changed so that it's more, they're detecting people, and they go after people, and they don't care about non-human animals anymore. So there you could say that this one gene makes Aedes aegypti bite us.
Yeah, isn't that cool?
Prefer us.
Yeah, that's right. Certainly contributes to it. And actually at that meeting, when we were last in touch in New York, the meeting on biology of genomes, woman named Lindy McBride, who was in Leslie Vosshall's lab and now has her lab at Princeton, was talking about related work that she's doing there. And really fascinating how in this case, the behavior of who these mosquitoes are picking to bite kind of impacts then on how the viruses are being spread or how they're working as vectors.
Too bad we couldn't get Leslie. Maybe we can get McBride on instead. Hey.
Yeah, Lindy's great. I'll keep an eye on that. She would be a great guest. Yeah.
All right.
Yep.
You're next.
Okay, here we go. Dear TWiEVO, Nels and Vincent, Here in Berkeley, California, it is 55 Fahrenheit, 13 Celsius. And in the past day and a half, we've had a glorious 1 inch, 2.5 centimeters of rain, something like 3.5 to 4% of our normal annual total, which we have not seen for several years. While listening to the great TWiEVO 382 with Ed Chung and Nels as guests, toward the end in a letter, the comb jelly came up, its anus having just been discovered. There was general, that's interesting. But to my surprise, neither Nels or Vincent commented on the evolutionary significance of the comb jelly. It appears to be a rare instance of complex biological elements having been created in parallel with the mainstream of evolution and not captured and reused, as opposed to the central retrovirus of TWiEVO 382, which seems to illustrate the parsimony of evolution. If there is something close to what I need, I will grab it and shape it to my needs rather than reinvent it. With apologies to Vincent for such anthropomorphizing. I snipped this from Evolution, You're Drunk. DNA Studies Topple the Ladder of Complexity by Amy Maxman. And there's a link here to this. I think this is a book a couple years ago, which is a popular science commentary on Ryan et al., the genome of the ctenophore. I'm going to just skip the genus and species and its implication for cell type evolution, a paper in Science from 2013, which seems to capture this key point. And here the quote, "Late last year, the animal evolutionary tree quaked at its root. A team led by Joseph Ryan in evolutionary biology who splits his time between the National Genome Research Institute in Bethesda and the SARS International Center for Marine Molecular Biology in Bergen, Norway, analyzed the genome from a comb jelly, a complex marine predator with muscles, nerves, a rudimentary brain, and bioluminescence, and found that the animal may have originated before simple sponges, which lack all those features. If comb jellies evolved before sponges, the sponges might have lost the complexity that the ancestor uniting them in comb jellies possessed. Or that ancestor, the ancestor of all living animals, had the genes to build brains and muscles, but did not form those parts, and neither did sponges. If this is true, then comb jellies deployed the genome they inherited to build a brain, nervous system, and muscles independent of other animals. There's some support for this possibility. A unique set of genes seems to underlie comb jelly's muscles. Both hypotheses run counter to scenarios in which organisms evolve to be increasingly complex. In one, a complex nervous system and muscles were lost in the sponges. In the other, the sponges had the genetic capability for complex features but stayed simple, while a more primitive group, the comb jellies, acquired brains and muscles that help them chase down prey. Furthermore, the idea that complex parts like a brain and nervous system, including nerve cells, synapses, and neurotransmitter molecules, could evolve separately multiple times perplexes evolutionary biologists because parts are gained one at a time. The chances of the same progression happening twice in separate lineages seems unlikely, or so biologists thought. Traditional views are based on our dependence on our nervous system, says Ryan. We think the nervous system is the greatest thing in the world, so how could anything lose it, he says. Or it's the greatest thing in the world, so how could it happen twice? Anyway, it seems that a TWiEVO that looked at this general issue, and maybe this specific research, would be interesting. This is William, or goes by Bill, from Berkeley, California. Yeah, wow. So a lot in here. And I think this actually, Bill is putting his finger on a really important and active area of thinking and research along the lines that we're aiming to consider and tackle on TWiEVO. I think the evolution of the nervous system is absolutely fascinating. I think that there are some of these views that he mentions that are sort of more traditional views that are starting to get re-examined and turned over in a sense. So this idea that there could be multiple origins of the nervous system. I think there's increasing interesting evidence for that. I'd also just make maybe one or two quick comments about how we think about sort of what's primitive and what's modern, or what's complex and what's simple, to take, at least what I try to do is take a little bit of an agnostic approach to that. So even though we often think of, let's just say, like budding yeast, because it doesn't have a brain, as primitive, since our ancestors split a really long time ago, hundreds of millions of years ago, we've actually had just as much time or gone through, if anything, yeast have gone through more generations than we have since our ancestors split. And so in that sense, I think of yeast, as being quote unquote just as highly evolved as we are. It's just that the sort of outcomes of drift and natural selection have just kind of led to a different niche. And so I think I try to caution, I think, some being a little bit careful about what we think about as advanced or a quote unquote higher organism, that I think we often can kind of throw a little bit of, sand into the gears of actually thinking, I think, productively about how evolution plays out and happens. So the idea of a living fossil, I'm not crazy about, because if it's still alive today, how could it be a fossil?
Yeah.
It's had just as much time to evolve. It's just come to sort of a different location or with different things have been selected on. And I think that applies to things like the nervous system as well. And things like even more complexity, the definition of intelligence, what that means. Maybe a better measure for success instead of having a brain is how much biomass your species, accounts for, in which case things like insects are really outgunning us, even though we're sort of busy trying to make, many other species extinct as humans are doing kind of crazy things. But anyway, thank you, Bill. I think we will definitely be picking up this idea of how, and of course, how like complex things evolve. I think that's often has been a tripping point for kind of the science curious public, and also these clashes between evolution and religion sometimes, in particular, these ideas of intelligent design where you say, oh, look at an eye, this thing is so complex. How could just, Darwinian evolutionary processes lead to something like this? And there are increasingly some really cool examples of how even something that we think of as complex as eyes show up in critters that are only a single cell, and are reacting to light in really sophisticated ways. And so these are sort of kind of starting to fill in the missing links, or have for actually many years filled in these supposed missing links. There's also, I think, related to this, and we should definitely get onto this topic, is the evolution of multicellularity, which gets at some of this. And there's some really great ongoing work from labs like Nicole King at Berkeley in Bill's neighborhood there as well. And so, we'll stay tuned. We will definitely be thinking about some of these ideas.
I don't know, Nels. We're going to have to increase the frequency. Better get tenured. How many more years you got to
keep you posted? So we're actually just starting that process now, and it takes about a year to. We'll see how it plays out. But the wheels are slowly starting to spin on that. Yeah.
We could go to twice a month.
We'll see what happens.
Nels is being cagey.
That's right, exactly.
If you got paid, maybe you'd do it, right?
Now we're talking. No. Look, this is really, I will just say, Vincent, I am having a blast doing TWiEVO.
Good.
Once a month, and we'll see. We'll see where we go from there. I would love to increase, but I have to.
You got a lab to run. I know. Yep. All right, just a couple more here.
Sure.
Jeff writes, please put TWiEVO on Stitcher. By the way, I'm an American computer programmer living in Medellin, Colombia, city of eternal spring. The weather here is always the low 80s during the day and low 70s at night. All right, you requested, I did it. We are now on Stitcher, which is another way to get your podcasts. Cool. Can you take the next one from Adam?
Sure. So here's Adam. Dear Dr. Racaniello and Dr. Elde, recently I came across an interesting concept, but I wasn't sure how to explain it in terms of molecular biology. Many animals possess certain instinctive behaviors, such as baby turtles running straight to the ocean after hatching, nest building by birds, cuckoos pushing other eggs out of the nest, and avian seasonal migration. While I can appreciate the evolutionary advantages of such behaviors, I wonder how these behaviors became instinctive in the first place. Was nest building once a learnt behavior, then became an instinct? If so, how are such complex behaviors passed down genetically? Was a transition from certain learnt behaviors to instinctual gradual? And how is this reflected in the changes of animals' genetic and epigenetic makeup? Thank you for sharing your knowledge and expertise on so many platforms. Greetings from sunny Manchester, UK. Adam. I think we're starting to get a theme here, Vincent, in this really kind of, I think, widespread interest in complex behaviors and the genetic basis of that, and the evolution of complexity in behaviors. And all of these examples, the sea turtles, I've seen this actually many years ago in a marine biology program. Just as the hatchlings come out of the eggs and then make a straight charge from the beach into the ocean, where they're under the pressure of many seabirds that are just there to pick them off. Most will pick off most of them, and then sea predators as well. And so, that's like to me, a beautiful example of a genetic conflict that then has these sort of really strong outcomes, at least from an intuitive level. Same with all, like the cuckoos pushing out other eggs, this idea of egg mimicry, or tricking another bird species to take care of your young. I mean, what a crazy conflict, and just sort of this, another example of that certainly has a genetic basis as well. Yeah,
I'm sure we'll be moving into this in future TWiEVO.
Yeah. And I guess it goes back to the answer to the previous question, is like, it's been really hard to get a genetic foothold on this. I think the Hoekstra lab, some of the Stickleback work that have made these really daring forays into doing this and are actively doing so. We're still working on it. There's also like some of the stuff I really love and hope my lab might actually transition into at some point is microbial control of host behavior. Mm-hmm. And, I think these are like really cool examples of how a lot of them are in insects so far, where they've been best characterized, but how a virus can infect a caterpillar and change its behavior to start climbing up a tree, such that as the virus is kind of replicating like crazy and basically dissolving the caterpillar from the inside out, it will spread farther because it's forced the caterpillar to climb higher, and that will disperse the virus. This is the one of the classic examples, and there have been a few genetic ties to that. A mimic of a hormone that might make the caterpillar more active, I think might be the strongest clue currently. And so to me, this is like actually microbial control of host behavior, where it might be sort of these simple tweaks of the host in these cases might be, I think, is actually a great avenue for getting some genetic foothold on this, because in these cases, it's basically just following or trying to learn how the microbes do this in order to learn something about the complex behaviors that we do all the time. And I think it gives a really kind of cool and possible experimentally tractable window into some of the behaviors that can otherwise look really complex. So stay tuned.
Yes, exactly what I was going to say. We've got lots of time left.
That's right.
All right, our last email is from Cheryl, who writes, "Please, gentlemen, I beg you, create podcasts more frequently." I've turned many of my colleagues on to the microbe TV programs, and they've all commented on how they love them all, but wish you'd do more frequent evolution podcasts. We would also love to have some evolution topics that could be used in the high school classroom to supplement class lecture. Thank you to all the TWi crews for the fantastic podcasts. And Cheryl is a world history teacher at a high school in Racine, Wisconsin.
Yeah.
I think a lot of the things we cover would be great for discussion in high school. And, the teachers could listen to our discussion, take notes on it, and then present it to the class as a way of, learning about the paper without having to dive through it on your own.
Yeah. I mean, that's certainly one of the challenges, right? Is as we're trying to explain this stuff, in a lot of cases, it's pretty complex. And so what level are we pitching it? And so it would be nice to get some feedback, actually. Are we, connecting in a way, what's clear, what's not clear, as we kind of continue to try to improve and do the best we can. But speaking of podcast frequency, I mean, I need to turn this back to you a little bit, Vincent. So your schedule with in the TWiEVO empire, with multiple in the family of podcasts, and then as you're adding these other things, I mean, my goodness, it seems like you're doing more than a full-time job at this point with all of the podcasting. It's really impressive and exciting to see.
Well, I really like it. And at this point in my career, I feel that I can make more of an impact doing this than laboratory work, although I have a small lab and I have a person who's very excited. And I love that. But I dedicate a lot of time to this because I just love it. I just absolutely love having these discussions in all different fields. And I'm now trying to raise money to get some technical help because it's now at the point where I have to do a lot of production, and I'd rather be recording podcasts instead of doing production.
So
I'm hoping I can raise enough money through advertising. I've also just started some crowdfunding efforts to hire at least a part-time producer to help me do this.
Yeah, that's a great idea. And what are the details on that crowdfunding that you've started?
So I've put up a page at microbe.tv/contribute. Mm-hmm. So I've done a couple of different things. First, you can go to patreon.com/microbetv. There's a link at the Microbe TV page. At Patreon, you basically subscribe to a creator. You become a patron of a podcast. You can give $1 a month, $2, $5, $10. You can give us $1,000 a month if you'd like.
Now we're talking.
And then we'll get you on one of the shows, or we'll come to your hometown and do a live show right in front.
Yeah.
So that's one way that gives a repeated donation, and a number of people have already taken that up, and it's great. I'm very flattered that's over a dozen people have decided to support us.
Oh,
but for people who just want to give us a chunk of money now and then, you go to microbe.tv/contribute, and I've got a button there where you can use PayPal or your credit card to send us some money. And, $1, $1 million, whatever you'd like to send, it's all appreciated. I don't want to make, it's not for me to make money. This is to try and, do some more traveling. So when people, so if someone said, hey, can you and Nels come to Florida and do an evolution at TWiEVO, I could say, hey, Nels, I got money. We can go there.
Cool.
Hire a production person and so forth. So that's what it's all about. So I must say I did miss May. I didn't post a TWiEVO in May, which was an oversight. So
we have an episode that needs to.
Yeah, I'll do it tomorrow.
Cool. That sounds good. Yeah, and just to say, I think I agree with you, Vincent, on all fronts there. And any support we could get would be so appreciated. And one of those, if you can forgo a fancy cup of coffee, even that would be really cool.
Cup of coffee a month.
Yeah.
If all of our listeners gave $1 a month, if 50% of our listeners gave $1 a month, this would make a big difference in what we can do. And I understand if you can't do it, that's fine. But for those of you, you could be the patron of the arts. You could be the Medici family.
Now we're talking. I love it.
All right. Let's wrap up with some picks. Nels, what you got here?
Picks of the week. So I was inspired, as I mentioned before when we had Jim on, about this idea that insects might be hermaphrodites. That is like, a single individual, both male and female, could self-fertilize. And so I just did a quick internet search last night, and the top hit was actually a fellow whose name already came up this episode, Ed Young, a great science writer, one of my favorites. And just as a side, I think Ed has a new book that's just about to spring out, or has, on microbiomes, sort of the microbes affiliated with us, called I Am Multitudes. I think I'm looking forward to checking that out. But anyway, so the link, my pick of the week, and the link is a article that Ed wrote in his blog, Not Exactly Rocket Science. This was, I think he's moved on. This was for Discover magazine, and he's actually had a couple of iterations since then. I think he was with National Geographic for a while, and then he's currently with The Atlantic, in terms of his kind of his recurring writing about emerging stories. But anyway, he did this one a few years ago on the complicated life of the cottony cushion scale insect. And there's a beautiful picture of it. It's really kind of disgusting. And this discovery that while most, as we were speaking about earlier in the episode, while most insects are male and female and undergo sort of reproduction through sperm and egg, that these scale insects have actually evolved to be hermaphrodites. And it's even more kind of bizarre and weird the way the male actually fertilizes its own daughter, so to speak. And so anyway, take a peek at the link in the article if you want to read about some bizarre and sort of fascinating biology when it comes to scale insect sex.
Nice. It's very cool.
Yeah.
I didn't realize insects could be hermaphroditic.
Yeah, and I think, as we said, this is probably certainly the exception rather than the rule, and it's probably super rare. But it kind of raises this idea. So the kind of interesting, I think Ed did a nice job of sort of balancing two different sets of researchers. One group that thinks this is a result of evolutionary conflict, basically where the hermaphrodites gain an advantage in a conflict between the sexes. So this is sort of another area that I'm sure we'll delve into, but is genetic conflict over sort of sexual reproduction, so investments by males versus females. This is all something that's been a kind of a foothold for natural selection to have these kind of really pivotal or kind of strange outcomes, one of which might be going from male-female to hermaphrodites, which is cropped up in many systems, including all the way out to the insects here. Or whether this actually is a more kind of a symbiotic or almost like kind of an advantage where it's not a conflict, but it's sort of almost altruistic. And so anyway, if you want to dig in to this sort of weird, cool biology, this is a really nice example of it. Yeah. How about you, Vincent?
Right.
What's your pick of the week?
Well, my pick is a new website or publication called Undark. You can find it at undark.org. It's a foundation-supported digital publication of the Night Science journalism program at MIT. And it's basically science journalism presented in a very nice way. If you go to their homepage, they have a series of panels with different stories they can click on, and a lot of interesting writers are working here. And they even have a podcast. And I thought it looked kind of neat. A number of people have been talking about this. A friend of mine, Dave Tuller, who's a science journalist at Berkeley, is going to be writing for them. And so I thought it would be good. Our listeners would be interested in this. One weird thing I noticed, I was poking around in the About section, and they write, Undark is not interested in, quote, science communication, unquote, or related euphemisms, but in true journalistic coverage of the sciences.
Ah, interesting.
I don't know what that means. How can you not be interested in science communication?
Well, maybe it's just the idea that it should just be part of communication, not that that our thinking about science or our conversations or the outlets for science are just part of communication in general. I mean, that may be something, this is a little off base, but it's kind of funny how we talk about in science, we talk about alternate careers sometimes. And
right.
when probably the alternate career is sort of what we've done with our careers, which is to stay in academics, whereas most people have gone on to actually probably careers instead of alternate careers. And maybe, I don't know, maybe that's what they're trying to capture, that this is just a part of communication as opposed to sort of like holding that out as science communication and trying to integrate that more closely.
Well, I mean, I don't know. Do you think you and I do journalistic coverage of the sciences? I don't think so.
No, I don't. I think we do something closer to just sort of sharing our kind of curiosity and interest in research that's unfolding and coming up almost in. Yeah.
I mean, I always call what we do science communication, and I don't know why it's euphemism, or they make it sound weird in a way. Now, I would have written that we don't do science communication, we do journalistic coverage of the sciences. I think that would have been the way to phrase that. I just think we're not interested in science communication would mean they would never listen to TWiEVO, which I find very offensive, because you got to hear Nels talk about evolution. It's great.
Oh, we're doing what we can. This website looks interesting, though. I'm seeing
it is very
links
interesting
here. How many women have Zika? Depends.
Yeah.
Stuff. Really neat links. Yeah. Cool.
Check it out. Right. And listen to our listeners, get some picks in. We don't have any today.
Yeah.
Get them in. And this TWiEVO will be at iTunes, microbe.tv/TWiEVO. You can also find it at Google Play Music. And we would love to get your contributions. As I've said, go to microbe.tv/contribute. And if you have questions or comments, send them to TWiEVO at microbe.tv. You can find Nels Elde at cellvolution.org. He is also on Twitter as Earlybird. Thank you, Nels.
Thank you, Vincent. Fun as always.
I'm Vincent Racaniello. You can find me at virology.ws. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.